All right, let's just jump right in with a statement that probably sounds like, I don't know, total scientific heresy, especially if you're in biomedical research. Ready? Here it is. All of the models we use to study disease, from cells in a dish to mice in a lab, they're all wrong.
Okay, hold on. Before you completely write off decades of scientific progress, just stick with me for a second. This is not some cynical takedown of science. Not at all. It's actually a really profound insight into how discovery really works. It's one of those ideas that once it clicks, it completely changes how you look at pretty much every single breakthrough.
So that's what we're going to do in this explainer. We're going to unpack this one single powerful idea. And it's funny, the author of our source material, Alessandro Esposto, he actually admits he loves dropping this line on his colleagues, just to see the shocked look on their faces before he explains what he really means. So let's get into that explanation. So how can something be wrong, but also right? To really get our heads around this paradox, we're actually going to take a little detour away from biology for just a minute. We're going to borrow an idea from a totally different field, mathematical modeling and physics. Trust me, it makes this whole concept crystal clear. So, what is a mathematical model? Well, think about a weather forecast, right? Or maybe a model of the solar system. It's basically a simplified version of reality. It takes just a few key factors, a few parameters, and uses them to try and predict what's going to happen. It's not reality itself, of course, but it's a really, really useful sketch. And this right here is the core idea. It's a total paradox, right? On one side, a model is always wrong. Why? Because it's a simplification. It can't possibly account for every single atom, every single variable in the entire universe. But on the other side, it's always correct, as long as it works, as long as it gives us the power to predict things with some level of confidence. If it helps us understand the world, hey, it's doing its job. And you can see this play out perfectly in the history of physics, right? Galileo's early models of motion, super useful. But then Newton came along, and his models with universal gravitation were even better. But then came Einstein, who showed that even Newton's universe was incomplete. See the pattern? Every single one of those models was wrong in some way, but each one was an absolutely massive, game-changing, and correct step forward. Okay, so with that idea in mind, let's bring it all back home to the, let's be honest, messy and super complicated world of biology. We're going to take a quick tour through some of these imperfect models that have, believe it or not, led to some absolutely incredible discoveries. So first up on our tour, we have the DT40 chicken cell line. Yeah, you heard me, chicken cells. Specifically, they're lymphoblast cells from a chicken, and they're used all the time in cancer research. And I bet I know what you're thinking, because it's what everyone thinks. Chickens? We're using chickens to study human cancer? Really? And you're not alone in thinking that. This quote here, "Are we trying to cure chicken cancer?" That's a real question from a real lab meeting. I mean, it's a totally fair, sharp criticism that gets right to the heart of how absurd it seems, right? We're talking about these floating, immature bird cells, not a solid human tumor. How on earth could this possibly be a good model? Well, here's the secret. These little chicken cells have a kind of superpower. They're unbelievably good at a process called gene recombination. And what that means is that scientists can go in and edit their DNA really, really easily. This makes them the perfect tool for asking super specific questions, like how a gene like BRCA2, the famous breast cancer gene, how does it actually work on a mechanical level? So yeah, they're the wrong model if you're trying to replicate a whole tumor, but they are absolutely the right model for answering that very specific genetic question. Okay, next up, let's look at probably the most common, most recognizable model system in the world. Cells growing on a flat plastic petri dish. I mean, this is the bedrock of modern cell biology. But let's face it, our bodies aren't flat, and they're definitely not made of plastic. And boom, there it is again, another zinger from the real world. A referee for a scientific paper once wrote, "Cancer occurs in vivo and not in a petri dish." It's basically saying, hey, your model is way too simple. It's nothing like a real living thing. And you know what? On the surface, it's a totally valid point. But to just dismiss the petri dish like that, that's ignoring like a century of progress. This simple, quote-unquote, wrong system has given us an absolutely incredible amount of foundational knowledge. I'm talking about the basics of how cells divide, how they talk to each other, how they eat. It's this simple setup that has allowed us to isolate and truly understand the fundamental machinery of life itself. And this whole principle of using imperfect models, it's not new at all. In fact, if you look back, some of the absolute biggest victories in the history of cancer research were won using systems that, by our standards today, seem almost, well, comically wrong. Get this. Way back in the early 1900s, a guy named Theodore Boveri made these huge leaps in understanding how cancer even starts by studying cell division in fertilized sea urchin eggs, sea urchins. I mean, talk about a distant relative. And then, a few decades later, Otto Warburg figured out fundamental things about cancer metabolism just by looking at slices of tumors in a dish. Simple. Wrong, and yet so right. These models literally changed everything. All right, now it's time to talk about what might be the most famous, the most important, the most controversial wrong model in the history of science. I'm talking about HeLa cells. So the story behind HeLa cells, it's famously complex, and we have to acknowledge the serious ethical issues right up front. These cells were taken from a woman named Henrietta Lacks back in 1951, without her consent. That's the bad. And then there's the wrong. Biologically, these cells are a total mess. They have something like 80 chromosomes instead of the normal 46. And yet, there's the good. As the researcher John Masters put it, so much of what we know about basic human biology today depends on these cells. From the polio vaccine to cancer research, these profoundly wrong cells have been absolutely indispensable. Okay, so we've seen all these imperfect yet incredibly powerful examples, and it all kind of boils down to one central, critical question. The kind of question that scientists in the field are debating all the time. So if chicken cells are wrong, and sea urchins are wrong, and petri dishes are wrong, then which system is the best model system? What's the right one? Is it a mouse? Maybe a primate? Which one is the winner? And the answer, well, it's as simple as it is profound. The truth is, it depends. It completely depends on the question you're trying to ask. There is no single best model. The best model is simply the one that helps you answer your specific question, while being fully aware of its limitations. It's really just about picking the right tool for the right job, and knowing that every single tool has its flaws. And this quote just sums it all up perfectly. All models are wrong, all techniques are limited, all experiments are somehow biased, but collectively, they can inform us. See, that's the heart of it. Science isn't about some heroic quest for one perfect experiment that unlocks all the secrets. It's about building knowledge, piece by piece, from a whole collection of different imperfect sources. It really helps you see that science is this big, passionate, collective debate. It's a bunch of scientists all using their own wrong but incredibly valuable systems. So maybe the next time you hear about some big discovery made in fruit flies or yeast or whatever, instead of asking if it's the wrong model, you can ask a better question. What question was it the perfect model to answer? Because that's where the truth comes from that big, messy conversation, where every wrong model has something truly right to say.